Welcome back, everyone. Today we have a very special video in which we're going to use the concept of incomplete lineage sorting to demonstrate common ancestry between humans, chimps, and gorillas. And if you don't know what that term means, incomplete lineage sorting, don't worry, we're going to talk about it in detail. So basically what we're going to be doing today is comparing two models with strong emphasis on the first one, but we'll talk a little bit about the implications for the second one. So the first one is the model of common descent. This is the evolutionary model. This is the model that all of science converges on as being the true one. And that's that humans, chimps, and gorillas in this case all share a common ancestor with one another. Alternatively, there's the creationist model, the model from Genesis, right? And that's that humans, chimps, and gorillas do not share a common ancestor, and that any similarity between them, whether in their genes or their morphology, is simply a reflection of what they call common design, okay? But what we're going to do today is we're going to look at the models that we apply to common descent and see if the model of independent origins can make sense of what we predict and what we actually observe in nature. So the first thing that we need to talk about is the genome as a mosaic, okay? The genome doesn't have just one topology or just one pattern of ancestry. The reason for that is because of recombination. Recombination breaks up associations between any given lineages and shuffles them around every generation, okay? So that for any given position within the genome, you can have a different ancestry. All right, so here's our hypothetical genome where the different colors represent each one of the recombination blocks, and then the separation event between the two colors, that's our recombination breakpoint, okay? So going left to right, we can see the tree topology is changing as we move down the genome, all right? Going from humans, chimps, gorillas, what we think of as the true tree, to gorillas, humans, chimps, humans, etc. All right. Now, under a model of common descent, we can use some very simple mathematics and actually predict what proportions we expect to find these discordant trees within the genome. Okay. And importantly, this model completely relies on common descent being true. It relies on humans and chimps actually sharing an ancestor before they do with gorillas. And that any of the discordance that we see in the genome is an emergent property of the fact that ancestral polymorphisms can persist into descendant lineages. Okay, so let's look at first what is a species tree. Okay, this is a new concept that we're going to introduce. Species trees are representations of true patterns of species relationships. Okay, here we've seen it in this kind of blocky background. Humans, chimps, and gorillas. Note that the size of these species trees normally reflects like effective population size. I didn't draw that to scale here. I just made them large so that I could embed gene trees within them. Now, another important thing is that species trees are never observed directly. Okay, species trees are inferred from patterns of gene tree proportions. Okay. And this is because species trees aren't written anywhere in the genome or written into the natural history of living things. They are emergent properties of the way gene trees sort in lineages. Okay. The next thing to bear in mind is the nature of any three-taxon tree. Okay, so for the tree humans, chimps, gorillas, this is a three-taxon tree, and there's three possible topologies that we can get. Two of them are the false topology. Okay, so humans, chimps, gorillas is one, humans, gorillas, and chimps, and then gorillas, chimps, and humans. Now here I've listed three ways to get the wrong tree, and I'm going to show you what I mean by that in just a moment, but only two of them actually have different topologies than the true tree. Another thing to remember is the time interval separating each splitting event is very important when we're thinking about species tree models, okay? For the interval between the splitting event of humans and chimps and gorillas, we're going to call this interval T1. Okay, and T1 is going to appear many times, and it's a super important variable to bear in mind as we go forward. So now let's actually throw a species tree or throw a gene tree into this species tree to see if we can kind of start to understand how this process works. So we sample a human, a chimp, and gorilla, and we sequence one part of their genome, just one gene, and we reconstruct their topology. Okay, and what we get actually matches the true species tree: humans, chimps, and gorillas. And we've just embedded it here within the species tree. And note how this happens, how this occurs, is that we trace this lineage back from humans, back from chimps, and that they coalesce, importantly, during the interval of T1. Right? This is during the interval after the splitting of humans and chimps, but before the splitting off of gorillas. All right? And so we have right here the coalescent event. Anytime you get a coalescent event during this interval, you have the true species tree. Okay. Now, if the coalescent event happens outside of this interval, it may or may not generate the true species tree topology. But if it happens within this interval, you will always get the true species tree. And then finally, back here where humans, chimps, and gorillas are all in an ancestral population, you get the final coalescent event during interval T2. Okay, so now let's go over here to this little equation and let's talk through how this is working. So we're calling the red trees type 1. This is our 1 tree. So this is the probability that we get type 1 tree is equal to 1 minus the exponential of negative T1. Okay, so what this means is this is the probability that we see this tree given the probability that coalescence has not occurred during interval T1. That's what this second term here represents, right? So one thing to note is that as T1 gets smaller and smaller, this variable here gets larger and larger, okay? As T1 gets longer, then this variable becomes smaller. And the way you can think about this is very simply that if this interval is short, right? If T1 was much shorter than what I've shown here, then the probability that humans and chimps coalesce during that interval is smaller, right? Because there's not as much time for them to coalesce during that interval if it's really small. If it gets really, really long, then they have lots of time to coalesce during that interval before they get to interval T2. Hopefully that makes sense. And so that's what this probability is showing us here. So it's all reliant on the length of the interval T1, okay, to what the probability of getting this tree is. All right, now let's look at a second topology that is exactly the same as this one, but actually doesn't have a coalescent event during T1. All right, so that's the orange tree that we're showing here. Now note that humans, chimps, and gorillas all coalesce during T2. And T2, remember, is the interval in which they're all in the same population. Humans and chimps still find an ancestor before they do with gorillas, right here. But it just happens to be that they're all within the same population. So they're in this ancestral population before these lineages coalesce with one another. So that generates the same topology. The branch lengths end up looking a little funky, right? Because you get longer branches to humans and chimps than what you do when the interval is in T1, obviously. And the probability is different. Okay, so the probability that we get this tree, which is a tree that includes what we call deep coalescence, because the coalescence happens back here in T2 instead of in T1, that probability is a third of e to the negative T1. Now, remember I said previously that there were 3 possible ways of getting the wrong topology. And by the wrong topology here, what we really should say is there's 3 possible ways of getting deep coalescence. Okay, getting a coalescent event that doesn't happen in T1, but instead is happening in T2. This is one of those ways, one out of those 3 possible ways. All right, so it just happens that it generates the same topology, despite the fact that it is technically a deep coalescent event. Okay, so now let's look at the 2 trees. Or, well, before we look at those 2 trees, let's make a simplifying thing so we don't have to keep up with this second topology. So note that we can just add these 2 proportions together, or these 2 probabilities together, and since we're only interested in trees with this topology, and we're not so much interested in branch lengths at this particular moment, we can just put these together, and that sum now gives us the probability of getting the correct species tree topology. Okay, irrespective of whether deep coalescence occurred, we have the human, chimp, gorilla tree. That probability is now 1 minus 2/3 e to the negative t. All we've done is we've added the 1/3 e to the negative t to this, right? So now it's 1 minus 2/3 e to the negative t. That's the probability that we get this topology. All right. Now let's look at the two topologies that don't match it. All right, so just like before, we have a scenario of deep coalescence. Okay, so there's no coalescent event happening in T1, it's all happening in T2. Chimps, gorillas are coalescing first, back here during T2, and then humans coalesce a little bit deeper. All right, that gives us this deep coalescent looking tree, chimps, gorillas, humans, and that probability is just the same as the other one. It's 1/3, because it's another one of 3 possible ways. E to the negative t. And then finally, the other possible way is where humans and gorillas, excuse me, coalesce first with chimps as the outgroup. This one gets a little messy because of the way humans and gorillas are laid out in this tree, so it looks like there's a crossing here. But what I'm really trying to show is humans coalesce right here, and this is the lineage to gorillas, and then chimps coalesce here after humans and gorillas. Again, so this is our type 4, so now we've got type 1 and 2, type 3, type 4, and these two probabilities, again, are exactly the same as the probability of human, chimps, and gorillas in a deep coalescence framework. Okay, so what are the implications of this model? I know that was a lot of information, but let's see if we can boil this on down to a simplistic couple of implications. So first is that species tree topologies, given T1 is not very small relative to the population size, as we talked about before, the species tree topology should be the most prevalent gene tree. You can see that right up here, right? This value should always be greater, given that T1 is not too small, should be greater than either of these values, okay? So this should be our most common tree, given our model. Secondly, discordant trees, these two, 3 and 4, should occur in roughly equal proportions. Because again, the probability of getting either of them is exactly the same, right? So they should be less than the number of topologies that show the species tree, and they should be equal to each other, right? That is what our model predicts. Now, let's take this model out for a spin. Let's see how it actually works in a study that sequenced a bunch of different organisms, humans, chimps, gorillas, orangutans, etc., built gene trees across the genome using 20 some odd thousand genes and see how it turned out. So this is a study from 2007 in Molecular Biology and Evolution by Ebersberger et al., mapping human genetic ancestry. They note here in the abstract that for about 23% of our genome, we shared no immediate genetic ancestry with chimpanzees, which means that we coalesce with a different primate before we do with chimps, okay, for 23% of our genome. All right, and so let's look at these 23,000 sequence alignments that they had across these different primates and see what the proportions look like. Do they recapitulate kind of what we expect to see? All right, so up here at the top, I've brought over our little toy models with the red, green, and purple trees. Remember, this is the true tree, or the true topology. This is one discordant, and this is the other discordant. And remember, again, these should be roughly equal, right? This is a table from their study. Here's the topology: human, chimps, gorillas, humans, gorillas, chimps. I've colored the boxes by the topologies up top. And then along this column, this is every single gene, or every single alignment they had. This is for every single gene, and this is for every single exon, okay? Over here, I've included a pyramid that's showing just like the spread of all maximum likelihood trees that they had, where you can see kind of a thickening up here in the true humans, chimps, gorillas, and then a thinner spattering of trees down here. All that you're seeing here in the middle, these are the different alignments that had like low phylogenetic signal, which means that there weren't enough mutations within those trees to get good posterior probabilities. So all the genes that I'm showing you here in this table had a posterior probability greater than 95%, or the tree reconstructions were highly confident. So for our most common species tree, this represented 76% of all the trees, or our most common gene tree, I should say, not species tree. 76% of all the trees were humans, chimps, gorillas. So they matched this topology, right? Which is kind of what we expect. The other two topologies are green and purple ones. The green one occurred at 11.46%, the purple one 11.39%. This is also true if you look at genes, 10.42, 10.17, and exons, 10.21 and 10.05. So, just as our model predicted, the two discordant trees occur in roughly equal proportions across the genome. And this is pretty incredible, right? This was a mathematical prediction based on a model of common ancestry in which discordance arises solely due to ancestral polymorphism. And we get the exact proportions that we mathematically derived to, and that we expected to see. Okay, so in summary, a model of common ancestry, as I was just saying, generates testable predictions about observable gene tree proportions across the genome. Again, remember that the genome is a mosaic, has lots of different lineages across it because of recombination, and so using common ancestry as our model, we can predict what those gene tree proportions should look like. When we apply this model to humans, chimps, and gorillas, it matches our expectations under our model of equivalent discordant gene tree proportion. So we exactly recapitulated what we expected to see. Now, I haven't talked a whole lot about the independent origins model because I don't have a strong intuition about how they would explain this. So two questions that I have for any creationist or ID proponent or person that thinks humans, chimps, and gorillas don't share a common ancestor of whatever religious background you have. What is the explanation of this pattern from your model? What do you expect to see? How do you explain the patterns of discordance across the genome and how they match exactly a model of common ancestry? And what predictions does an independent origins model make concerning gene tree proportion? Is the idea that the discordant gene trees are reflecting the percentages of where chimps and gorillas are more similar than they are to humans and vice versa? And if so, why are those proportions exactly equal? Can you point to exactly equal number of morphological characters that are shared between humans and gorillas and chimps and gorillas? And is that the prediction? And if so, can you give some examples as to how that actually is found in nature? So yeah, anyways, with that, I hope you learned something. I hope you found this interesting. If you have any questions about incomplete lineage sorting, deep coalescence, or just like coalescence in general, how this model works, etc., feel free to drop me some questions in the comments, and I'd be happy to chat through it. Thanks for watching, and have a great day.